Well, I'm really happy to be hosting Dr. Claudia Meisrimler and Sophie Eccersall to discuss the recent PLOS ONE Lab Protocol article entitled Phytophthora pluvialis maintenance, spore production, and detached needle assays, and their associated detailed protocol in protocols.io. Claudia received her PhD from the University of Hamburg in Germany in 2012. Her early research focused on plant iron and phosphate starvation, investigating enzymes involved in redox reaction and reactive oxygen species production. In her last postdoctoral position at Utrecht University in the Netherlands, she began focusing on plant pathogen interactions. Claudia achieved tenure in 2019 and started her own research group at the University of Canterbury, Christchurch, New Zealand. Her current research focuses on the interface of plant pathogen interactions and immunity. She has a keen interest in signal transduction and signal crosstalk, with a particular focus on omised effector proteins that modulate plant signaling and fast response mechanisms in host plant, including calcium and ROS signaling. Sophie has a Bachelor in Science with a double major in Chemistry and Biology from the University of Auckland in New Zealand and a Master of Science with distinction from the University of Canterbury, also in New Zealand. She's currently doing her last year as a PhD student, working towards a postgraduate degree in cellular and molecular biology, supervised by Dr. Meisrimler and Professor Renwick Dobson at the University of Canterbury. Her project is based around identifying and characterizing the targets of RXLR effector proteins in Phytophthora pluvialis. Sophie has a keen interest in pathogens, particularly Phytophthora, and figuring out how they work. She has expertise in a range of techniques, utilizing cellular and molecular tools in combination with biochemistry and bioinformatic methods. Thank you both for carving some time to talk to me.
No problem at all.
To kick off, can you briefly explain what the protocol is about, how you use it, and why it is relevant?
Certainly. So the whole basis of the protocol is to be able to induce zoospores from Phytophthora in order to then use those in further experiments. So you can then do things like detached needle assays, which is what we delve into in the protocol itself. And then you can further use those detached needle assays in other experiments as well.
And to help us understand the significance of the development of this protocol, can you elaborate a little bit more on what was the state of the art when you decided to go ahead and do it? So were there similar assays or assays that could do the same? And if so, why those assays didn't work for you?
So that was really the driving factor for it. When I started working with Phytophthora, I didn't really have much of a background in there. There are numerous protocols out there, but I think it's one of those things where you're working with a biological organism and they're not all the same. They don't all behave the same. And so you can be given a protocol and it simply just may not work. So it was really, really important to work through that. And that was something that Claudia really encouraged me to do. I was so used to, you know, when you're in undergraduate and you're doing laboratory work, those experiments have been done hundreds, thousands of times, and they work. And then suddenly when you get a protocol that doesn't work, you know, as a new postgraduate student, you often think that you're failing and that you're not working. And why isn't it working? But Claudia said to me, you know, you've got to realize that things like this aren't always done, and there are going to be problems, and you do have to troubleshoot them. So that was really the driving force for me to try and get this to work and be reproducible and replicable, which is obviously very important.
And delving a little more on the protocol itself, can you share what are the most critical steps, those in which users might most likely mess up?
So patience is key with the protocol. So, you know, when you're having the initial like mycelium growing on a plate, for example, that's fine. That's fairly easy. You can let the organism do that. That's not a problem. But when it starts to come to the sticking point where you're actually trying to get the spores to induce and mature, you do need to be patient. You need to remember that every single strain is different. And it may be the same species, but a different strain. And if you are trying to push something too quickly, it's simply not going to work. So you do have to allow yourself to have that time. If you don't allow yourself to have that time, you'll simply end up frustrated and with no spores.
And I think that the next question, the response to the next question might be really useful for the potential users. As you were developing the protocol, was there anything that you tried that didn't work in the process of optimization? And because it didn't work, it didn't make it to the final version of the protocol itself?
I would say yes. There was a lot of different media types that I tried, different concentrations of the media, you know, so a standard is, you know, like a V8 media or a carrot-based media, things like that. So I really did play around with the concentrations to see what was going to be the best and gave the best outcome. And also there were some things along the lines of different types of water. I know that sounds, you know, we think of water, it's water. But that was another thing that was, you know, you couldn't use, for example, sort of sterile deionized water because you just simply wouldn't get the same result. So those things are important to think about. And yeah, those were things that just simply had to be cut.
And before you mentioned that, you know, different strains, even of the same species, they develop at different rates. So I want to know if either before or since the time of publication, how robust is the assay for different strains? And if so, how much of tweaking you have to do in the protocol itself, other than probably just wait different times when you try different strains?
So the biggest thing is just the wait times. But it is important to really have good strain maintenance to make sure that those strains are growing well and they're healthy. Because if you leave them for a while, sometimes they can just stop. And I did have one strain that I was working with. I was working with 5 different ones, and I getting a little bit frustrated. I was thinking, why isn't this working? Why isn't this working? I talked to another person who is a specialist in this field, and this person acknowledged, she goes, oh no, that one doesn't work anymore. It simply doesn't work. So I think it's important that, you know, you get to know the strains that you're working with. I know that might sound a little bit strange. It's, you know, it's Phytophthora, it can't talk back to you. But I have had it continue to work. I've also had it continue to work on some other Phytophthora species as well, which I've just sort of played around with. This wasn't anything that was going to be in the protocol, but you know, it was worth trying. It was worth seeing if we could get other strains, or other species, I should say, to work.
So I just wanted to kind of like say that it's quite important to keep pathogens that are plant pathogens to kind of like, part of the maintenance is to put them on a host and not keep them on the plate continuously. So because if you keep them on the plate forever, they get very much used to this kind of like perfect life because they get everything they need from the plate and they get lazy. So you have to teach them again, they have to work for actually, yeah, staying alive. And so that is kind of like the part where Sophie did needle assays and then transfer back from the needle onto the plate because they have to work. And yeah, so this is kind of like really important. I've seen for many Phytophthora species in the past that if you keep them just on a plate for longer than 4 to 6 months, they stop infecting. And ultimately, we want to do infection assays to learn about the interaction between pathogen and the host. And if they don't do their job properly anymore, then it's kind of like a bit of a lost cause. So this is quite important, I think, kind of like looking at the overview of Phytophthora species, that kind of like is something we've seen with multiple kind of like different Phytophthora species.
So in addition to the patients that you need to have with different, trying different strains, it is the maintenance of the pathogen themselves and challenge them regularly.
Yes.
With not just providing everything they need on a plate, but making them work.
Exactly. Yeah, I think the challenge is part of what a pathogen is in general, but with these Phytophthora species in particular, yeah, if you pamper them too much, they just don't perform really well.
Thank you so much. Very insightful. And changing gears a little bit, when you started, Sophie, your research and you struggle to get protocols to work, you optimize this beautiful protocol, but then you went a step ahead and then you decided to share it with the community and have it peer reviewed. So what was the motivation for you to do both things? First of all, share it publicly in an open access repository, and then have it peer reviewed as a formal standalone methodological paper.
So my biggest motivation was, there was the initial sort of selfish motivation of getting things to work for myself, but then I wanted to be able to share it for anyone who was in a similar position to me, who was coming into an area that maybe wasn't their specialty and they were trying something new. I wanted them to have a really solid protocol that they could see step by step what they should be seeing on the plate in the lab, rather than just sort of looking at words and going, I think that looks like how it should. Actually being able to see what that was, a tangible thing right there. So they could go to their microscope and go, yes, all right, I'm not quite at that stage yet. I can wait a little bit longer. And that way people would be able to work through things a lot easier, I think. And that's what I wanted. I wanted other people to be able to use it. It would to be user-friendly, and also for people to contact me if they need to, to open a conversation about these things, and to have it peer-reviewed as a standalone, manuscript. It's one of those things that it makes me feel like it was justified, that there's other scientists out there who have looked at this, they have said, yes, this is sound, this works. And for me, that's really important. I want other people to be able to use it. And then if someone else has peer reviewed it, they're saying that, yes, this is a good thing. This is worthy to be out there. We should be using this. So for me, that was really a key driving factor there.
And can you tell us how the feedback that you received from your peer reviewers shaped the final version of the protocol?
So that was really, it was helpful because they, when you have something peer reviewed and you are so used to reading something and proofing something over and over again, you can get a little blind to it. And so when a peer review, comes in and they say, hey, have you actually thought about this? Or we would really like it if you could include this. And you go, oh, yeah, I didn't even think of that. So it's you know, another little extra layer of security. It makes the story more complete. It can make the story more robust. That's you know, my sort of point of view from it. Yeah.
And ultimately, that's how the qPCR made it into the protocol.
Yeah.
Because originally we didn't have the qPCR kind of like in there. And the other student working on the qPCR data, Leanne Vinson, already established kind of like one of our effectors as kind of like a go-to control. So we just asked her to contribute this part. And that was quite interesting and nice, because basically it allowed another student to come on board, which is not usually kind of like we had to actually explain why we have someone else kind of like coming as an author. But I think there was a reasoning behind it, and that was based on a peer review comment that it would be really nice to have actually a qPCR to actually validate the quantity, or to quantify basically the infection if you have if you do the needle assays. And that was very it was a totally valid question and request, and so that made it in because of the peer review process.
And it's satisfactory when the peer review is a constructive process and makes the story more complete.
Yeah, I totally agree. And I have to say that our peer reviewers for this, and also the editor from PLOS they were very nice. They were very patient and very friendly. And so then it's a really nice process for students as well. And we all know that can go very different. Peer reviewing can be a bit of a harsh process, especially for students that come new on board for this system. So it was very polite and very friendly, and that was really good as well.
I'm happy to hear that. And actually, my last question, could be a little bit speculative at this point, because your paper is very, very young. It has just 2 months since publication. But what do you think has been or could potentially be the impact of this paper for both of your careers at very different stages, like one very, very young, another one a tenured professor?
I'm quite excited about it because, you know, this was something that I put a lot of time and effort into, obviously. But I think that, and I hope that it can help establish me as a specialist in the Phytophthora field, particularly as Phytophthora pluvialis, which is what I primarily work on, is actually spreading outside its usual geographical locations and outside of its normal host range. So I anticipate that there will potentially be more scientists working on it. And if there are more scientists working on it, they're going to need to be able to maintain it, use it in the lab. So I'm hoping that, you know, people can utilize the protocol when they're doing their work. So I'm, you know, almost using it as a little bit of a springboard, but I think it's just a really nice base for me to work off.
And yeah, for me, I mean, I haven't worked with Phytophthora pluvialis before Sophie started with this project. So I worked with Phytophthora capsicum, which is kind of like probably the easier, or it's definitely an easier Phytophthora to work with. So for me as well, it's kind of like, established in the field of Phytophthora pluvialis work, but also kind of like we're going to use in the future this protocol or we're already using it with collaborators to get some new research going in kind of like treatment of this pathogen. And industry for kind of like some of the host trees are also gonna try to make this into a very high throughput screening for basically breeding purpose of pine trees. So in a way, it's kind of like it was a good starting point. And generally, I hope that it will be useful to people that start working in Phytophthora pluvialis. which is spreading, as Sophie just mentioned. So in UK, it's kind of like it just went through the whole UK within 2 years. It's now basically everywhere in the United Kingdom. And so in a way, I think it's going to be a very problematic pathogen in the future. So if we can fast track what other people are doing in the lab, that would be quite handy, I would say. That would be really, really good, because it's important research, at least in my point of view.
And before we finally wrap up, is there anything that you would like to mention that we haven't touched so far in this conversation?
No, not from my point of view. I think we covered pretty much everything from the protocol.
Yeah, I think my only thing I did mention before was that what I really like about this sort of thing that we're doing here with the interview is that, you can put a human face on a paper, and it really, we have our motivations for doing science when we are doing manuscripts and papers, etc. But we have to be, certainly kind of clinical about these things. But being able to chat about it like this just, puts that more human sort of touch on it, and just sort of shows that there's a person behind it. This is their reasons. And, I encourage people, if they do have any, questions or comments about anything regarding this, to contact me, talk to me. It's my passion. I'm happy to talk about it. So really thrilled to be able to share that information today.
Well, thank you. Thank you so much again.
No, thank you. Thank you for having us.
Yes, thank you.